Hello everyone, my name is Yuchien. Today I'm going to talk about adaptive significance of light and food for kleptoplastic sea slug. First, we have to know what is kleptoplastic sea slug. Kleptoplastic is a symbiotic phenomenon. A symbiotic means a close or long-term biological interaction between two biological organisms, depend on each other for survival. This kind of sea slug eat algae for food. They can steal chloroplast from algae and keep the chloroplast inside their body and use for photosynthesis. The kind of this research called Elysia atrovirides. The order is Cercoglassa, and they eat Codium flagell for food. And it can retain chloroplast inside their body about 1 to 3 weeks. This is Codium flagell. The result will mention reproduction traits, so I'm going to introduce. First, the number of egg masses. We can look at the picture. The egg masses looks like a colorful flower, and inside it, there has a large number of eggs. Second, shell length of larva. Larva means immature form. Third, reproductive investment. It is the number of offspring multiplied by offspring size. The research wants to figure out the effect of light intensity, low or high, and food availability on survival rate, body weight, and reproductive traits. On April 2016, they collect this sample at Mukashima, Hiroshima Prefecture, Japan. And why they choose the time at April? It's because Elysia atrovirides were most abundant at April, and they before they begin to lay eggs. Okay, they do the experiment is a long-term experiment. They spend 15.9 weeks, and long-term experiments spend nearly the whole reproductive life of Elysia atrovirides. And long-term experiment more likely to detect the effect of light and food on the survival rate than the short-term experiment. This is our experiment treatment. We can look at the upper left is weak light and unfed. Upper right is weak light and fed. Bottom left is strong light and unfed. Bottom right is strong light and fed. And we can look at the weak light. The brightness of weak light is 1 micromolar per square meter per second. Strong light is 30 micromolar per square meter per second. Okay, I divided the prediction into 3 states. First, when Elysia atrovirides can obtain energy from food and photosynthesis, under strong light has better adult survival and growth, greater reproductive performance than weak light. Second prediction, Elysia atrovirides cannot obtain energy from food. In starved condition, will have bad effect on survival rate, body weight, and reproductive trait. Then we look at our result. We can first look at the 2 lines that I circled. We can see the 2 lines are all in fed condition. Red one is strong light, blue one is weak light. Then we can see under the fed condition, strong light has better survival rate than weak light. According to second prediction, we look at other 2 lines. Red one, they are all in unfed condition. Red one is strong light and blue one is weak light. Then we can see these 2 lines are all lower than fed condition, these 2. So then we look at this figure. X label is number of weeks, Y label is survival rate. The survival rate, we can look at this, put the same color together to see. Red line has higher than strong light, full of the red icon is higher than hollow icon. Then we can know the survival rate remain higher under feed than unfeed condition. Full of red icon is feed and hollow icon is unfeed. And the same as blue line. The full of blue icon is higher than hollow icon. Then we look at the body weight. X label is number of weeks, Y label is body weight. Then we look at the line that I circled. They are all in feed condition. Red icon is strong light, blue is weak light. Then we can get the result is under the feed condition, strong light has better adult growth than weak light. According to the second prediction, we look at other two lines. Red one is strong light and blue one is weak light. These two lines are all lower than WF and SF, so we can know under the unfeed condition, the body weight is lower than under the feed condition. Then at this discussion, we just look at the SF, strong light and feed. ELISA heterogeneous require both light and food to attain the maximum performance. We can look at this red line are the highest at these two figures, survival rate and body weight. Then we look at the reproductive trait. First, we look at the same two lines. According to first prediction, we can know under the feed condition, strong light has more egg masses than weak light. We can see this red line are higher than this blue line. According to second prediction, we look at the other two lines, W and SU. They are all in unfeed condition. These two lines are higher than feed condition. Then we look at the shell length of larva. X label is for treatment. Y label is shell length of larva. We look at the right side of the figure. We can see the strong light is higher than weak light. So we can know that under the feed condition, strong light has a longer shell length than weak light. Next, according to second prediction, we look at the left side. We can see on feed condition, the shell length is shorter than other two. The next, under the feed condition, we can clearly see the red one is at a strong light, is higher than weak light. Second prediction, we can see these unfeed conditions are lower than feed condition. So we can check the first prediction. It can obtain food and photosynthesis. It means it have feed condition. Under strong light has better adult survival and growth, greater reproductive performance than weak light. Then we can check the second prediction too. It cannot obtain energy from food, but sour condition will have bad effect on survival rate, body weight, and reproductive trait. It's the last prediction when ELISA heterovarieties cannot obtain energy from food and photosynthesis. The effect of light condition on the reproductive trait are expected to be small. Then we look at the only reproductive traits. The number of egg masses, we look at the unfeed, W and SU. Under the unfeed condition, the effect on the number of egg masses is small. We can look at these two hollow icons. They didn't have significant difference. Okay. The number of egg masses is higher under unfeed condition here in the first 1 to 2 weeks. And we enlarge our figure here. It's because This is Elisa Echeverria's special life history tactic. It can use resources for egg laying before they start to die. Second prediction, we can look at this figure. X label is number of weeks, Y label is number of egg misses. Under the strong line and unfeeded SF, we can see individual continue to lay eggs here after 4 weeks. In the natural condition, it will lay egg at May, just after 4 weeks in April. Then we look at shell length of larva. X label is forced treatment, Y label is shell length. Under the unfed condition, we can see it didn't have significant difference. The last is reproductive investment. Reproductive investment is the number of offspring multiplied by offspring size. X label is for treatment, Y label is reproductive investment. Under the unfed condition, look at the left side. We can see the effect of light on reproductive investment is small. They didn't have significant difference too. So we can check the third prediction. In conclusion, both light and food are needed to exhibit positive effects, not only on survival or body weight, but also on the egg number and size. Thanks for listening.
The slides are easy to read, and the speech is steady and slow and steady, and which is easy for audience to follow the talk.
Hello, and your prediction and resource discussion are clearly to follow, but maybe I forgot or I don't hear you why you want to do this research. And in the end, I hope you can organize a table to explain the situation under different variables. Maybe we can easy to understand in your conclusion.